Now for translation. So we have had multiple steps in this process. We've had DNA. We've taken DNA and built RNA from it. That process was transcription. I think of transcribing as writing something over. If you're writing something over, it's still English to English, nucleic acid to nucleic acid. What we're doing now is translation. If you translate something, you're taking it from one language into another language. So we're going to now use a nucleic acid language to build a protein. That's a great way of remembering the difference between transcription and translation. We're now building a protein, we're building a new language out of this code, these directions of letters. So, the first thing that happens is a ribosome has two separate parts. It has a small subunit and a large subunit. The first step is that my mRNA transcript, so I'm going to have that cap and tail, and remember it's got a poly-A tail at the end of it, that will actually attach to the small subunit. When it attaches to a small subunit, transfer RNA molecules start to be attracted in to the codons. Every 3 letters of mRNA is a codon. So A, U, G is usually what we call the start codon. That AUG would attract a transfer RNA molecule in. So over here I'm going to have a transfer molecule come in, and the letters need to be complementary on the tRNA molecule. So that would be U, A, C. And this is attached, remember it has sort of that funky clover leaf shape, and it carries with it methionine. Methionine is the amino acid that is specific to this transfer RNA molecule, and that comes into this first place. As soon as that comes in, the larger subunit of the ribosome fits together. So we have a large subunit here that comes into play. The large subunit, if I redraw my ribosome again, the large subunit has a P site and an A site for these transfer RNA molecules to come into place. It also has an E. E stands for exit. When something moves to the exit, it's going to be able to leave the ribosome. So here, I'm actually going to make this a little longer because it looks really short right now. So I'm going to make my mRNA transcript a little bit longer. I have initiation, the first step. My first transfer molecule has come in. It has brought with it its own amino acid. The next thing I need is the next codon. So each 3 letters of mRNA is my codon. And every 3 letters that correspond to it of transfer RNA, remember this is my tRNA molecule, this is called an anticodon. And they always are complementary to each other. So I am going to have, let's say, CGA. So CGA, what pairs up with it? That would be a G, a C, and a U. And that would be attached to the next transfer RNA molecule. And that would carry a different amino acid on it. When these 2 amino acids are really close to each other, in the P space and the A space, they form a bond. Hopefully you're saying in your head right now, what type of bond forms between 2 amino acids? What's it called? Peptide bond. So this right here is a peptide bond. And remember, the goal of ribosomes is to build proteins. If we're taking amino acids and hooking them together, we're building a protein. So what happens next, this whole tRNA molecule has done its job. It has brought in methionine. So this piece right here can now leave. It can go to the E and it can exit. And what happens is this one now slides over. The methionine, it has let go of the methionine, so the methionine is attached to this amino acid. And now we have another new spot. So let me draw what's going to happen. Here is my ribosome. Here is my amino acids that are being hooked together, one at a time. So far I've brought in 4. transfer RNA molecules, because you can see 4 amino acids hooked together. And as soon as they're done, the transfer RNA is left back to the cytoplasm to pick up another amino acid just like it had before that's its right match. So in the P site, I'm going to draw my little transfer RNA molecule. That had this one. In the A site, I've got another. And here's the newest peptide bond to form. This will continue over and over and over again until, do you remember how there was that poly A tail on the mRNA? Eventually, and here's all the part that's already been used, eventually I'm going to reach the end. Or I might reach what's called a stop codon. So the 3 letters and the 3 letters might code for a stop. And what happens is a release factor comes in here. So I'm going to have these slide over. And the newest thing to come in here is what we call a release factor. It does exactly what it sounds like. It cuts this off. So now what I have is this strand of amino acids. That is the primary structure of a protein. So here is my polypeptide. It has several amino acids all hooked together by peptide bonds. It's very specific because the order of the mRNA codons determined which amino acid which would come in sequence. So the sequence of these amino acids is very specific for the job it's going to do. Right away, it's probably either going to take on that alpha helix or beta pleated sheet patterning. That was my secondary level of structure for a protein. And then, let's say it's an alpha helix, it can fold over on itself, maybe form some of those disulfide bridges or positive-negative ionic interactions, or maybe even this is very hydrophobic and this is very hydrophilic. So it will fold. Folding is the 3rd level of structure. And if you have multiple chains come together to build an overall large molecule, that is a quaternary protein, probably some big globular enzyme. This one that I drew here looks like hemoglobin. So the process of building proteins happens at the ribosome. There are 2 subunits that come together. When they come together, there are 2 spots for the transfer RNA molecules to come into place. Each transfer RNA molecule carries an amino acid. So if I imagine this is my P site and this is my A site, here I am. I'm the first one, so I come right into the P site. Why did I come here? My anticodon, so imagine these little 3 lines coming off the bottom of me. Those 3 lines match up the 3 letters on the floor. The 3 letters on the floor are from the mRNA transcript. I have my amino acid. In comes another transfer RNA molecule. Its pattern matches what's there, so its anticodons match the codons. It's carrying its amino acid, and since now we're so close together, forms a peptide bond. I've now done my job. I would go out the exit site, and the one that was in the A site slides over to the P, and then another one comes in, carrying its amino acid, and that is how a protein is put together. It will be really helpful to you to go to my wiki and to view where it says extras. There are some animations. They're like a little bit science fiction, whatever. There's do-do-do-do music in the background. But as each molecule comes in, I think it will help you really visualize it and be able to put this together. Please ask me questions in class if there's something about this that you're not understanding.